right, everybody, welcome back. Hope you had a great Thanksgiving. You know, as bad as my voice sounded a week ago today, I never actually got that sick. My wife said it was a good thing that my voice didn't work. So, you know, it was a happy Thanksgiving all around. Everybody had a great time. But welcome back. And today's the day I'm going to start talking with you about the regulation of gene expression, how genes get turned on and how genes get turned off. At the beginning of the class, we were talking about what makes something alive, right? Weeks ago, months ago now. And one of the big things was the ability to respond to stimuli. Well, turning genes on and turning genes off is a really big part of how it is that living things respond to stimuli. And today, for the first time, I'm going to get down and dirty with you to talk about just how it is that prokaryotes pull that off. And I'll talk specifically about how E. coli pulls that off, but it'll apply to all prokaryotes. And then on Thursday, I'll give you some insights in terms of how it is that you and I, as eukaryotes, do that, and the extra challenges that we have that prokaryotes don't have in doing those same tricks. To get to that spot that I want to be today, though, where I'm talking with you about how genes get turned on and turned off, I want to talk with you about two other things just quickly, and then we'll get to the good stuff right after that. So last Tuesday, I fought my way through a lecture about viruses. Those of you who were here know that we had some trouble with recording that particular lecture. Those of you who weren't here may be wondering what happened with that particular lecture. Because we had trouble recording it, what we did is for the Tegrity recording, instead of using that one where I could barely speak and where there were also recording problems, we just put up last year's lecture, the equivalent lecture. So I don't think you'll miss a beat if that's what you go back to look to. In fact, I think you'll be better served if you listen to last year's recording as opposed to what I tried to do this year with in the throes of laryngitis. But, so that's all on the pilot page for the course. But I talked with you on Tuesday last week a little bit about viruses. We'll go through that really quickly here. The long and the short of it is the first viruses that were discovered and characterized were out of tobacco plants, actually. The tobacco mosaic virus has that particular distinction. It's the first virus to have actually been discovered. What the researchers did is they took sap from an infected plant, no cells. They could look with a microscope and see there was nothing cellular that was present in the sap. They filtered it to make sure that there was nothing that was of the size of a cell that was present in that liquid. And yet that liquid from the sap that had no cells in it was enough then to give rise to an infection in an uninfected plant and convert it to an infected plant from which they could get more of the sap that had stuff that was too small to be a cell, yet still could give rise to an infection. And hence the idea that viruses are small, they're smaller than cells, and yet they have some of the characteristics we expect to find with living things. And that is they can make copies of themselves if they're given the right opportunities. So again, that's what was going on with tobacco mosaic viruses. I spent a little time last Tuesday talking with you about the fact that viruses all have two things in common. They've got a protein coat that we call a capsid that protects the genetic information on the inside of that capsid. That genetic information comes in, I'm tempted to say all shapes and sizes. Probably should be a little less facetious and tell you that it comes is a nucleic acid, but it comes as double-stranded DNA, single-stranded DNA, single-stranded RNA, and there are even viruses that use double-stranded RNA. So two things that all viruses have is a protein coat, that capsid, and then inside is nucleic acid. And viruses do come in a lot of different shapes and sizes. The simplest viruses are these rod-like structures. Again, you got your protein capsid with some nucleic acid in the middle. You get some of these cooler dodecahedral kind of Dungeons and Dragons kind of die kind of structures. This is one that has not just the capsid, but also has encapsulated the viral particles with some lipids to give it some extra protection and give it extra abilities to infect particular cells by fusing to the plasma membrane of an infected cell. And then you've got my personal favorites, the ones that look for all the world, like a lunar lander module. I teased you a little bit on Tuesday telling you that what's at the tip of these lander legs for these types of viruses, this is a bacterial virus that we're seeing a picture of here. It's bacteriophage T4. But what that virus is actually looking for are proteins on the surface of a cell that the tips of those legs can interact with and stick to. And they sort through cells trying to find the cells to which there are the right matching proteins on the surface, right? The virus doesn't want to infect a cell that it's not capable of overtaking and taking over and making more copies of itself with. And the way that this particular virus ensures that it infects only suitable host cells is by looking for specific proteins on the surface of those cells. T4 actually looks for a protein I'll be talking with you about later this morning, one of the proteins involved in lactose metabolism. And I'll remind you of that when we get to that point. But so there are viruses for you very quickly in a nutshell. Oh, okay. And I talked with you about how viruses are playing this game where their ambition is they want to have more copies of themselves when the world comes to an end than anything else. And there are a couple of strategies that you might take to do that. One of those strategies is just as soon as you infect the cell, make as many copies of yourself as you can and bust out and go find more cells that you can infect. A longer game and potentially a better strategy is that if the cell that's been infected is healthy, to hunker down inside of that cell and ride it out until that cell comes upon hard times, and then pull the trigger, make copies of yourself, bust out, and infect other cells. So the two strategies I've just described are a lytic sort of strategy, a lytic phage, as opposed to a lysogenic strategy, one where you lay in wait and then ultimately do go lytic when the time is right. I think I used the analogy rats abandoning a sinking ship, viruses abandoning a not so healthy cell. All right. So a couple words for you about viruses. I said I had two things I wanted to talk with you about before I got to the good part, the gene expression part. The other thing I wanted to talk with you about is just kind of the lay of the land of bacterial chromosomes, and also want to make sure that we're all pretty solid talking about plasmids. Okay? So I've said it how many times now? 10 times during the course of this class this semester that bacteria we expect to have a big old circular chromosome. I mentioned in passing at a couple points that, you know, when I was taking freshman biology, that was actually a hard and fast rule. But bacteria had a single circular chromosome. Now that bacterial genomes are being sequenced pretty regularly, pretty routinely, there needs to be a little bit of a modification to that rule. Turns out that many bacteria actually have more than one chromosome. Right? So they'll have two chromosomes typically. But the picture is still pretty good, that there's a small number of chromosomes, and those chromosomes are big old circles. E. coli has just one chromosome. It's a big old circle, 4 million nucleotides long. Okay, from beginning to end, in that circle, that's how many letters are needed to spell out the genetic blueprints that lets you make a new E. coli cell. Okay, one big old circular chromosome. Most prokaryotes, in fact, all prokaryotes, I believe, have, in addition to those big circular chromosomes, some smaller things called plasmids, extra chromosomal chunks of genetic information. Plasmids. Plasmids are circles as well, circular pieces of DNA, but they're much smaller, typically on the order of a few thousand to a few tens of thousands of nucleotides long in that circle. Okay? So they're smaller, and they're also dispensable. A bacteria doesn't have to have a plasmid in order to survive. It has to have that chromosome and the genetic instructions associated with it, but plasmids bring some extra sort of superpowers, if you will, to a bacteria that has them. Plasmids are typically where it is that the genes that confer antibiotic resistance reside. So you hear about superbugs, bacteria that are getting resistant to a lot of different kinds of antibiotics. Well, those antibiotic resistance genes tend to reside on plasmids. In the plasmids, these small circular bits of DNA can be passed fairly easily from one bacteria to another bacteria. Bacteria are pretty good at picking up those circular bits of DNA. And so if you have one bacteria that's a superbug, it becomes pretty easy for other bacteria in other bacterial species to pick up those plasmids and obtain their antibiotic resistance. All right, so plasmids, small extra chromosomal bits of DNA, not essential for the life of the bacteria, but again, they often have with them the coding sequences that are needed to give the bacteria some unusual or some special abilities that the bacterial chromosome by itself doesn't give. So the genetic information in bacteria is stored by and large, for the most part, on these big chromosomes, but there are these little sort of cassettes, if you will, these little plasmids that can be moved over from one bacteria to another. There's one special kind of plasmid, though, that I want to talk with you about for just a couple of minutes right now. And that special kind of plasmid is called the F plasmid. F is short for fertility. And it is the F plasmid that allows a bacteria like E. coli to have some kind of sex. Right? We've talked about the advantages that come from being able to reproduce sexually, to be able to break the association between very good genes and not so good genes, right? That recombination that comes from sexual reproduction. Well, bacteria have sort of a poor man's way of having sex, and it's associated with these F plasmids. Okay? I'm going to talk with you specifically about how E. coli uses an F plasmid in the next couple of minutes, but other prokaryotes have F plasmids as well. So this is sort of a universal theme for prokaryotes that allows them to have fertility. So let's talk a little bit about that E. coli chromosome. You know, I could imagine that this picture is fairly intimidating. It shouldn't be. There's not a lot to actually see here that you guys need to worry about. What you're seeing is actually a map of E. coli's chromosome. And it's not even a complete map. It's just a map that shows you some of the clusters of genes that we know reside on E. coli's chromosome. So here's a cluster of genes. It's LAM, and then there's an MKEFG. Right? So there's a set of 6 genes here. Those 6 genes, I'll tell you more about this in a little bit. These genes are part of an operon. So these are genes that code for proteins that have a related function, things that all need to sort of be firing at the same time in order for a metabolic pathway to work, for instance. And bacteria tend to cluster their genes together in this, I think I say in the lecture notes, an anal retentive way. They're very ordered. Genes with related functions tend to be located right next to each other on a bacterial chromosome. That's not the case for humans and for eukaryotes, but bacteria have genes with similar functions right next to each other as part of a single operon. Here's another set of 4 genes. These are involved with leucine, the amino acid synthesis. Right? So there apparently are 4 genes that you need, 4 proteins that you need to be able to synthesize leucine from scratch. They're cleverly named A, B, C, and D. They're all clustered together, and that's where they reside. This is the operon that I'm going to be spending a fair bit of time talking with you about today, the lactose operon. There's a set of 4 genes that are important, 4 proteins that need to be expressed in order to metabolize the sugar lactose. So what you're seeing here is just a circular map, right? It's a big circle, obviously, and it's supposed to let you see where along this circle clusters of different genes are, right? It's a map, in other words. How do you get a map like this? Well, today, the way you get a map like this is you use a sort of an inelegant brute force approach, you sequence it, right? You crack open the bacterial cell, you pull out its DNA, and you use chemistry to go across the whole circle and determine the order of every letter, G, A, T, and C, across that entire 4 billion nucleotide long circular genome. In the old days, and still as a prelude today before people do that brute force crack it open and sequence the darn thing approach, what would be done would be conjugational mapping experiments, okay? You guys know what the word conjugational means. You've heard of conjugational visits. We're talking here about sex. I've already tipped my hand and told you that sex in prokaryotes is facilitated by an F plasmid, okay? And so F plasmids can be used to determine the order of genes on a circular chromosome like this. And let me show you how. So here's a slide that shows you the general picture of what's going on with F plasmid conjugation. Check it out. You've got a cell here. It's got a chromosome within it. It's also got an extra chromosomal bit of DNA here. It's called the F factor, or an F plasmid. Okay? You know what F factors or F plasmids are good for? They contain the information for 27 proteins, a small number, right? Not a big deal, a few thousand nucleotides long, right? Those 27 proteins let the bacteria that have an F factor generate what's called an F pilus. An F pilus, for all intents and purposes, is a protein rod, a hollow tube that extends from the surface of the cells that have an F factor and allow that cell with the F factor to bump into cells that don't have an F factor and squirt their DNA into them. Phallic, right? You know, I mean, that's the right word to use to describe this. This F factor allows a bacteria to generate what's called an F pilus. And the F pilus is a hollow protein tube that allows the cell with the F factor to transfer some of its DNA into a cell that doesn't have an F factor. Okay? And so you can see what's happened here is this cell with the F factor and its F pili has hooked up with a cell that doesn't have an F factor, and it's going to start snaking its F factor DNA into the cell so that at the end of the process, what you've got is you've got the cell with the F factor that has squirted the DNA for the F factor into a cell that didn't have an F factor. Okay? Now this cell can break that connection and make its F pili of its own and go out and have sex with other cells that don't have an F factor. Simple, right? But this is about conjugational mapping. I'm only telling you about this, well, I'm telling you about this to give you a sense of what plasmids are about. Small things give sort of a superpower and extra ability to a cell that has them. But I really want to focus now on this idea of conjugational mapping. So this is conjugation going on here. We've had a transfer of DNA from one cell to another cell, but sometimes the F factor isn't hanging out by itself as a plasmid floating around free inside of the cell. F factors can also have a special ability. Most plasmids do not do this, but F factors can also get integrated into the host cell's chromosome itself. So instead of being a little circular red plasmid, independent of the big blue chromosome, it's possible for F factors to become linearized, all right, instead of a circle, become a line, and then integrate into the bacterial cell's chromosome so that the bacterial cell's chromosome becomes bigger, right, a little bit bigger. But instead of having just the bacteria's genes, it also has in its chromosome the information you need to make an F plasmid and F pili. So when that happens, now we're in a position where you can do conjugational mapping. Because it shouldn't be too surprising to hear that if you interrupt a bacteria that is conjugating having sex with another bacteria, if you interrupt them, you're going to disrupt the flow of genetic information from the one bacteria to the other bacteria. And so what folks can do, microbiologists do this in the lab all the time for conjugational mapping, is they'll get a bacteria, they'll trick it into integrating an F plasmid into the chromosome, they'll let that bacteria start to have sex with another bacteria, and then they interrupt that conjugation, and they look to see what genes got transferred over from the first bacteria to the second bacteria. The longer you let them go, the more genes get transferred across. And if you interrupt them every minute or so, right, you get a population of these guys. You get one population going for just a minute, another population for 2 minutes, another for 3 minutes. If you interrupt them at different times, you can see the order in which genes get passed from the one bacteria to the other bacteria. So what's happening here is this. Check it out. You got an F plasmid that's gotten integrated into a host cell's chromosome. All right. Conjugation begins. An F pilus got made. And the DNA that's getting snaked through is a copy first. Here's the F plasmid that got integrated in. The first thing that gets copied is this A gene. It goes around the circle of the chromosome, picks up the B gene, and then that gets snaked over into the new cell that hasn't seen this. And check it out. These are capital letters on this chromosome, and there are lowercase letters here. They're homologous genes, but they may be different alleles, different versions of those genes. And check it out, right? A little bit longer goes by, and now it's not just the A and the B that got passed over, but the C is getting copied, and it's starting to make its way through. And then something happens. Somebody comes in and separates the two, busts up that connection, and all that made it through, apparently, in this particular conjugation experiment was a copy of the A gene and the B gene. But because the B gene here is capital, and this one is lowercase, and this one's capital, and this one's lowercase, it might be possible to tell the genes that got passed over were first the A gene, then the B gene. If you do this experiment again, the next time you might get the C gene coming through. Do it again, you might get the D gene coming through, but they're always going to come through in an order that's determined by where that F plasmid became integrated into the host cell's chromosome. Could be for different integrations, right? If this had gotten integrated over here, they'd come across in a different order. Where the rubber I must be shouting too much in this class, right? Because I burned through batteries on this microphone more than I've ever burned through batteries in the past. Maybe it's just a very weak set of batteries that we're using in the biology department these days. But what's happening here is that for the last 15-some years, every time I've given a final exam for the Bio 1120 class, I've asked a question about conjugational mapping, and it's a little puzzle, and here's the thing. I think you guys can feel comfortable, you know what's up with conjugational mapping, if you can take the time and work your way through one of these problems. So I'm telling you, you're going to see a question just like this on the final exam. Take a couple minutes, make sure you're comfortable with how it is you determine the order of genes from this kind of data. And I'll walk you through it right now, and then we'll start talking about the regulation of gene expression. So here's the deal, right? I've got for you here information from 5 different strains of bacteria that have an F plasmid integrated into the host cell's chromosome. 5 different strains, 5 different F plasmids. Well, it's always the same F plasmid, but it's integrated into the chromosome in 5 different locations. Cool? So when the F plasmid is integrated into the chromosome, you call those cells HFR cells, high frequency of recombination cells. Okay? And they get arbitrary names. The first one must have been, Henry must have been the guy who came up with this one, right? He likes the letter H. He calls this particular HFR strain the H strain. And when they do conjugational mapping with the HFR strain, the first thing that gets passed through is something that's part of the F plasmid itself. That O is the origin for transfer. But then the next set of genes that get passed over with the H strain are the genes associated with threonine biosynthesis. That's what THR is for. So there's an operon, a set of genes that's involved with something involving threonine biosynthesis. The next operon that gets passed through is the leucine operon. The next one, AZI, TON, PRO, the LAC operon that I'll talk with you about in just a couple of minutes, the ADE operon. You don't have to know what those operons are about to do a conjugational mapping experiment. You just need to appreciate that these operons are getting passed over in a particular order. Okay? A different HFR strain, let's go down to this one here, 6. Got inserted, the F plasmid got inserted into a different spot on the chromosome. When conjugational mapping experiments are done, the first thing that gets passed over is something that's part of the F plasmid itself. But then you'll see right before the first operon that get passed through was the threonine operon. Here it's the isoleucine operon. Why? It's because the F plasmid's on a different location. It's big circular chromosome. The F plasmid here has landed in a different spot. And so we've got a different operon that's getting passed through. Isoleucine, methionine, THI, THR, leucine, this one, TON, right? Here's where the mapping comes into play. You're getting a look at the lay of the land on that chromosome just from the information here, but you want to really see the whole picture. You got to link together the information you get from multiple HFR strains. And you may have noticed that between HFR strain 6 and H, there's actually some overlap in the information that's come through. ILE, MET, THI, THR, this isn't the first time I've seen THR. THR is up here as well, isn't it? What comes after THR? Well, after THR is LEU. Hey, sure enough, I've seen that before. What's after LEU? AZI. I've seen that before. What's after TON? Here, I don't know. But from this one, from the HFR strain H, I know that after TON is PRO, then LAC, then ADE. What's before THR? Here, I don't know. But here, I know that before THR was THI, and then MET, and then ILE. And so you can start to see, if I take the information from 6 and H, I can start to get a bigger map than I would have been able to get if I had used the information from just H or just 6 by itself. Cool? So you can see there's sort of a puzzle that's going on here. Let's say I want to know what's on the other side of THI, or better, ILE. What comes before ILE? Well, check it out. Here's an ILE from HFR4. On one side is MET. Yeah, there's MET. I already knew that. And before MET is THI. I already knew that. On the other side of ILE, though, is MTL, XYL, MAL, and SER. So the circle's getting bigger. I'm getting bigger and better read all the time. Some of you may be a little worried that the order here is sort of the opposite. All that means is that in the HFR strain 4, the F plasmid landed in one orientation, and in 6, it landed in the other orientation. So you're reading off in different directions between 4 and 6, but you can use the information from these 3 HFR strains to get a better read across the whole circle. And by the time that you've used these other 2 HFR strains, what you'll find is you can make a map that's a big circle, goes all the way around and never really ends at that point. And what I can do to you, once you've made that circular map, and I think the only way to do this is with a pencil, writing it out on an exam, is I can ask you which of these 5 choices corresponds to the sequence of genes that you saw in the puzzle that you've deciphered in the circle that you've been able to make from the information with those 5 HFR strains. Okay? So that's what's up with conjugational mapping. Again, it gives you a ground truth, in a sense, of what you might get if you did an actual exhaustive sequence of the genome. If you sequence the DNA of a bacterial cell, you may be worried that they got shuffled around a little bit as part of the sequencing process. This tells you this is the actual order. We've seen some biology that confirms to us that this is the order of genes on this particular chromosome. One last time, I think you've heard me. You'll see a question very much like this on the final exam. This is just a chance for you to, roll up your sleeves, mess around, decode this puzzle. I don't think it should take you more than 5 minutes to work this sort of thing out. And if you can do that, I think you can be confident, okay, I know what's up with conjugational mapping. It's a sort of satisfaction for solving the puzzle. Okay, finally, right? We got all this chromosome stuff out of the way. We got viruses cleaned up and finished. Now I can tell you about how genes get turned on and turned off. Let me start by just emphasizing, sort of shouting out to you, how genes get turned on and turned off is, I'm inclined to use some profanity here. I'll try to stay PG-13. It's a huge deal for bacteria to be able to figure out how to turn genes on and turn genes off. It's a huge deal, okay? Let me impress upon you why it's such a huge deal. This is especially true for prokaryotes. It's less true for eukaryotes, and I'll tell you why on Thursday, but it's a big deal for prokaryotes. It costs a prokaryotic cell a lot of energy to make a protein, right? That's the bottom line. Let's talk about the numbers. Let me try to impress you with some of the numbers here. I'm sure that's what you're all excited about. Ooh, math, numbers, right? Think about it. How do you make a protein, right? If you're a prokaryotic cell, how do you go about making a protein? You got to transcribe the gene, right? In English, that means you make an RNA copy of the DNA sequence for the gene, okay? Making an RNA copy, doing transcription, isn't cheap. It literally costs 1 ATP worth of energy for every nucleotide you string together to make an RNA molecule. With me, right? Because what are the nucleotides that you're stringing together? They're ATP, UTP, CTP, GTP. That's what's getting strung together. You're burning the equivalent of 1 ATP for every nucleotide that you transcribe. ATP, it's the dollar bill of the energy currency of a cell, right? Bacteria are all about using their energy resources better, more efficiently than their competitors are. It all comes down to how efficient they are with their energy stores. I'm not making that up. I'm not exaggerating. That's what it's all about. One bacteria does better than another bacteria simply on the basis of how well it uses its energy reserves compared to something else. It's all about energy. Every nucleotide that gets transcribed costs you an ATP. How big is a typical messenger RNA inside of a prokaryote? Well, let me come at this from the other direction. A typical protein is a couple hundred amino acids long. Let's just, between friends, we'll make the math easy. Let's say 333 amino acids long. That's a little bit big for a protein, but it's not crazy big, okay? A couple hundred amino acids long. If it's 333 amino acids long, how many nucleotides long is that? 3 nucleotides, triplet codons for every amino acid, right? So 3 times 333 means the coding region was about 1,000 nucleotides long. To make the messenger RNA's got to be 1,000 nucleotides long if it's going to code for a 333 amino acid long protein. So a typical gene, for the sake of argument here, let's say it's about a messenger RNA needs to be made that's about 1,000 nucleotides long. And I've been trying to stress to you here, a messenger RNA that's 1,000 nucleotides long means the bacteria, when it's decided to make this protein, is committed to spending 1,000 ATPs. That's a lot of cash, right? 1,000 ATPs. That's like I'm asking you, dish me out 1,000 singles from your wallet or your purse right now. You're going to stop and think before you start, let's get the 1,000 single dollar bills laying out here. Right? That's a big deal. Right? That's a serious commitment. When the bacterial cell decides that it's okay to make this protein, at some level, there's a commitment to expend 1,000 ATPs. But that's not all. It also costs ATPs to string amino acids together as part of the process of translation, right? Because that's not the whole shebang. Making an RNA isn't all, that doesn't get you very far. You need the protein. To make the protein, you got to translate it, that messenger RNA, with a ribosome. And for every amino acid that you link together to make a protein, you should expect to spend 2 ATPs, one to bring the amino acid into place and one to charge the tRNA with the amino acid that's getting brought into place. It costs you 2 ATPs for every peptide bond that you end up making. So now we're at what, 1,600, 1,700 ATPs for one copy of a protein. But you're probably not going to make just one copy of this protein. Messenger RNAs can get used over and over and over again. You may make 100 copies of that protein. Whoa, now this is getting seriously expensive, right? Now we're talking about maybe 20,000, 30,000 ATPs that you've blown by deciding to express this gene, okay? Serious moolah. You guys were worried about my asking you to dish out 1,000 singles, right? Now I'm asking you to just, 20,000, 30,000. I'm confident nobody in this room has that number of singles. In fact, all of us together don't have that number of $1 bills in our pockets right now, right? This is a serious, big deal. There's like dollar bills. There's ATPs flying around like crazy for this particular process of making a protein. And you could argue that's not even the whole story, because those amino acids don't grow on trees. In some sense they do, but the bacteria has to make those ATPs, right? Not just ATPs, they have to make those amino acids. Amino acids cost anywhere from about 10 ATPs to about 70 ATPs to make. Glycine costs about 9 ATPs to make. Tryptophan costs 72 to 73, depending on the circumstances the cell is living under. There's an opportunity cost at the very least. If you're incorporating amino acids into a protein that you don't need, you got to make more of that amino acid for the proteins that you do need. Do you see why I say it's a big deal, right? I mean, we're talking tens of thousands, maybe 100,000 ATP commitment when a bacterial cell decides, oh yeah, I'm going to turn this gene on, right? It sounds like a light switch kind of thing. Oh yeah, what's the big deal? It's a huge deal, especially when a bacteria is living in this world that's so competitive, that's all about energy. You don't burn ATPs unless it's for a good reason, okay? Now, in the scope of things here, let me take a step back and tell you that the total cost to make an E. coli cell, right, from scratch, if you want to go from 1 E. coli to 2 E. coli's, the energy expenditure is measured in terms of about 1 billion ATPs, right? So making 1 billion ATP, if your bank account has $1 billion in it, if you're Bill Gates, right, it's not that big a deal. It's not as big a deal to Bill Gates to spend $100,000 on a nice dinner as it is for you and I, right? For you and I, $100,000 for a dinner, what is your problem? You're messed up. That is ridiculous. Don't ever even think about spending $100,000 on a dinner. For Bill Gates, $100,000, it's probably worth it. There's probably a diamond ring in the turkey stuffing somewhere. It'll be all right, right? I can handle it. It's not that big a deal. I read a story a number of years ago, but that, what's the smallest amount of money that you'll pause to pick up, right? You see, oh, look at that. There's a penny on the ground. People just walk right past pennies, left and right. You can do this experiment. Take some change, throw it on the ground, see if people, you'll see them notice, oh, look at that, there's a penny. And they'll walk right past it, right? How many of you will stop for a penny? Let me see a show of hands. A couple people, right? There are some people who will only pick up a penny if it's heads up, right? So there's that. So we can divide those people in half, right? How about a nickel? How many of you will stop for a nickel? Now, I'm warning you, at some point I want everybody to raise their hand. Right? So if you raised your hand for a penny, I sure hope you'll stop for a nickel. How about a dime? If you recognize the dime, will you stop and pick up the dime? How about a quarter? Right? So you guys are holding out for the big silver here. How about a 50-cent piece? Right? Oh, that's just because of the novelty. You don't see too many of those. How about a single? How about a dollar bill? Everybody will stop for a dollar bill, right? Some of you won't. Are there holdouts who won't stop for a dollar bill? Right? There are a couple. Probably nobody would pass up a dollar bill. If nothing else, I'll stop for the hope that it's actually 100 that's crumpled up in a way that makes it look like a dollar bill, right? Well, Bill Gates apparently, the average person at the time I read this story about 10 years ago, the average person won't stop for anything less than a quarter. The equivalent to Bill Gates, given his bankroll, it has to be a check, a personal check made out to him for $17,800 some dollars, right? So he won't. It's not worth his time. If it's not worth your time to stop to pick up a nickel, it's not worth Bill Gates' time to stop and pick up anything less than a personal check for at least $17,000, right? So, again, E. coli costs about 1 billion ATPs to make another E. coli cell. So that's the scope of things. But even still, 100,000 ATPs, even when you got 1 billion ATPs in the bank, that's enough to get Bill Gates' attention. It should be enough to get a bacteria's attention, okay? So the energetics of all this, turns out the accounting, the economics, just the bookkeeping of this turns out to be a big deal. And when I tell you then that E. coli, it's very much to the disadvantage of E. coli to express a gene that it's not going to get a return on its investment from, right? That's what it all comes down to. E. coli's got to constantly be making this decision. For every gene that it has, am I going to get a return on this investment? If I spend 100,000 ATPs to make this protein, am I going to get at least 100,000 ATPs back? Or alternatively, am I going to save at least 100,000 ATPs? It all comes down to that. If the perception of the cell is, I'm actually going to get 1 million ATPs back, that's a really good investment. Of course, let's spend the 100,000 ATPs, because I'm sitting on a gold mine here. I'm going to get 100,000 back. That's a great return on the investment. But if I think I may only get 50,000 ATPs back, bad idea. Don't spend the 100,000 if you might only get 50,000 back. It all comes down to that. So, E. coli has constantly got to be making this decision. Do I express this gene or not? And let me tell you, I need to back up just a second here, and I've been using some jargon, one word that every time I said it, I think, boy, I got to tell them what I mean. When I say express a gene, I mean make a protein with that gene encoded. Okay, so there's this gene expression process. It's the central dogma. You start by making an RNA copy, then you translate it into a protein, the whole shebang from beginning to end. Let's call that a shorthand word. Let's say that's gene expression. The gene got expressed. So if you make a protein, the gene's been expressed. Again, E. coli's got to be agonizing all the time for every gene that it has, and it's got a couple thousand genes, about 3,000 genes. For every gene that it has, it's always got to be doing this sort of miserly, Scrooge-like sort of thing. Oh man, am I going to get more ATPs than I spend when I express this particular gene? Okay? So let me show you a practical example of how this plays out. Okay? I've mentioned it a couple times already today in a few earlier classes. E. coli has something called the lac operon. All right? Lac, it's short for lactose. Lactose is a sugar. You may get that sense just from the name with the O's at the end. Glucose, sucrose, ribose, right? Sugars. Organic chemists name sugars and they put O's at the end of them. Lactose is a sugar. Okay? It's specifically milk sugar. So mammals make milk. There's a sugar, there's an energy source within milk. That sugar is lactose. Lactose happens to be a disaccharide, meaning that there's two monosaccharides that get strung together, glucose and something else. I'm drawing a blank on what the other thing is, but it's not a big deal, right? It's a disaccharide, glucose and something else. The something else gets converted into glucose, and the two glucoses from lactose get shunted through glycolysis, and for every glucose that you run through glycolysis, you're going to get, what, 30-some ATPs. Okay? If E. coli finds itself in an environment where there's lactose around, there's potentially some energy to be extracted, right? You can see how this could play out pretty well. If there's a lot of glucose around, it might be pretty cool. It might come in really handy for that bacteria, that E. coli, to metabolize the glucose, break it up into its two sugars, and then run those through glycolysis, and it's sitting on a pile of ATP. The more glucose there is, the more ATPs it can make. This could work out. Do you think you might get 100,000 ATPs? Depends on how much glucose there is, doesn't it? Okay? But it depends on how much glucose there is. If there's no glucose, you know what? It's a no-brainer. It's real easy to walk away saying, I'm not going to make the proteins that let me metabolize lactose. That's a bad investment. If there's no glucose, there's no prospect at a return on that investment. And so E. coli then has to be paying close attention to how much lactose is out there in the world. If there's a lot of lactose, let's make these proteins. If there's no lactose, back off, bad idea. The genes involved with lactose metabolism should only be expressed when lactose levels are high. Right? Obvious, logical, nothing particularly mystical or insightful about it. It just boils down to that. It makes no sense to make the proteins involved in lactose metabolism unless lactose is around. What are the genes involved in lactose metabolism? It turns out that there's three. Okay? They're cleverly named. French guys came up with these names, by the way. They got a Nobel Prize for figuring this out in the 50s. Monod and Jacob and Monod in English, Jacob and Monod. Every time I say that, I think both of them spin in their graves a little bit because they would be appalled at the English pronunciation of their names. But Jacob and Monod worked this out. They figured out there are three genes involved in lactose metabolism, named Z, Y, and A. Those are their one-letter abbreviations. The actual names for the protein encoded by the Z gene is beta-galactosidase. Okay? The actual name for the gene encoded by the Y gene is galactosidase permease, and the gene encoded by the A gene is galactoside transacetylase. A lot of times people will just call it transacetylase. Let me cut to the chase for you here. There are three genes. There are three proteins you need to have if you want to metabolize lactose. Here's what those proteins do. Beta-galactosidase, the Z protein. That's the guy that breaks the disaccharide into two monosaccharides. Pretty obvious. You need that to be able to metabolize lactose. In fact, it makes you may wonder, what else do you need beyond that? Well, there's two other things you need. Another thing you need is the protein that's encoded by the Y gene. The Y gene is what gives rise to the channel that lactose can pass through to get from outside the cell to inside the cell. Sugars, water-soluble, phospholipid bilayers, not so much. Sugars have a hard time working their way through. You need that protein, a transmembrane protein with a special job, only allows lactose to pass from one side of the membrane to another. It happens to only go down a concentration gradient. There's no active transport going on here. It's just a pore in the cell's membrane, a hole in the cell's membrane, and if there's more lactose on one side of the membrane than the other side, it lets that lactose pass through, simply by diffusion. The third protein, transacetylase, it's got the job of once it encounters lactose, it puts an acetate group onto it. And what that effectively does is it prevents the lactose from going back out of the cell the same way that it came in. Permease won't let acetylated lactose go out, so that stays inside of the cell, and it sets up a concentration gradient so that lactose on the outside can come in, but once it gets to the inside, it can't get out because acetylase has acetylated it. Okay? Pretty good strategy, actually pretty clever strategy when you stop and you think about how it is that it's working at a molecular level. But the end result is that if you make these three proteins, you'll be able to get lactose from outside the cell to inside the cell. You'll be able to keep it in the cell until you can bust it up and run it through glycolysis. Cool, right? Can you think of anything else you might need to get ATPs out of lactose? I can't. Apparently nature can't. Those are the only three genes that are involved in lactose metabolism. Okay? Those three genes all sit right next to each other on E. coli's circular chromosome. They're part of a single operon, a cluster of genes with a related function. Right? They're all involved in metabolizing lactose. They're doing three different things, but it makes sense to have those three genes hanging out right next to each other. It's a very nice, neat sort of level of organization, isn't it? And so these things are sometimes called structural genes. They're coding for proteins that are involved with the metabolism of lactose. They all share one promoter. I haven't said anything to you guys at all yet about what a promoter is. At least not that I recall. A promoter is a string of nucleotide sequences that an RNA polymerase will recognize or can recognize as being the start of a set of genes. Okay? Because what's going to make that RNA copy? RNA polymerase. RNA polymerase doesn't just make RNA copies of any old DNA sequences, it makes RNA copies of DNA sequences that correspond to protein coding regions, to genes, right? So the genes of the lactose operon have a single stretch of nucleotides that an RNA polymerase can recognize and say, oh, this is the start of a set of genes. That's called a promoter. Every operon has a promoter. Every operon has a string of letters, a string of nucleotides that RNA polymerases, as they're passing along, can stop and say, oh, look at this. This is a promoter. There must be some genes that could be transcribed just that-a-way. Not that way, but that-a-way. Okay? Curious minds have got to want to know, what's a promoter? Or beyond that, what are the letters? Well, the letters come down to this. The letters come down to, it's got to be a string of nucleotides that would allow an RNA polymerase to be able to confidently discern between what's the start of a set of genes and something that's not the start of a set of genes. Given the size of E. coli's genome, about 4 million nucleotides long, right? It turns out that about a dozen letters, about a dozen nucleotides is enough for E. coli to be able to say this is the start of a gene, that's not the start of a gene. Whenever it encounters at least 12 specific letters in a row, 12 specific nucleotides in a row, it can say, this is a promoter. If I don't find all of them, it's not a promoter. I'm not going to stop and start to make an RNA copy. Those 12 letters are clustered in 2 separate groups, which is sort of 3 separate groups. For something to work as a promoter, there's got to be an A. That's one of the letters. The A is the first nucleotide that the RNA polymerase will transcribe. Okay? So that's one letter that you've got to find. That letter, that A, the transcriptional start site, the first nucleotide to get incorporated into a new RNA molecule as it's being made, that gets a fancy name. Molecular biologists call that the +1 nucleotide. The first nucleotide to get transcribed, +1. It's always an A. The nucleotide that comes after the +1 nucleotide, you guys are way ahead of me, I'm sure. It's the +2 nucleotide. The third nucleotide to get transcribed, +3. The fourth, +4, right? You get it. It goes on for however long. It could be +3000, right? That's the 3000th nucleotide to be transcribed. For the computer scientists in the room, this is going to, and the hardcore mathematicians, what I'm going to say next is going to freak you out. It's going to, like, fingernails on a chalkboard kind of thing. This just isn't right, but it's the way molecular biologists think. What's the nucleotide that comes before the first nucleotide? -1, right? Computer scientists and mathematicians have a really hard time with molecular biologists' lack of respect for 0, right? But it goes +1, +2, +3, go in the other direction. It's -1, -2, -3. There is no 0. Okay? For those of you who are not mathematically inclined, you're like, yeah, what's the problem? Of course it's -1. But for the math people, that's a big deal. Move on. That's just the way we talk. Okay? So there are 13 nucleotides an RNA polymerase is looking for. One of those is at +1. Another set of 6 begin at -10. So -10, -9, -8, -7, -6, -5, -4, right? There's a cluster of 6 nucleotides that get called between molecular biology friends, that they get called the Tata box. You know why they get called the Tata box? Nothing sexual. Get your minds out of the gutter, right? It's because one of those is a T, -10 is a T, -9 is an A, -8 is a T, T-A-T-A-A-A-A tends to be what a Tata box looks like. So there's a string of 6 letters, 4 of which are T-A-T-A, and then there's another A-A after that, right? So RNA polymerases are looking for that -10 sequence, followed by the +1 sequence. They're also looking for something called a Pribnow box, named after the guy who first characterized it, that's found at -35. So a stretch of 6 nucleotides at -35, a stretch of 6 nucleotides at -10, 1 nucleotide at +1, you're at 13. That, when an RNA polymerase encounters it, an RNA polymerase, if it had a brain, it would stop it and would say, this is a promoter. To that direction, in that direction, must be the coding sequence for a gene I'm going to start transcribing here at that +1 nucleotide. Cool? So, back to this map of the lactose operon. You've got a set of 3 structural genes, open reading frames, that have the information you need to make beta-galactosidase, lactose permease, and lactose transacetylase. You've got a promoter that an RNA polymerase might recognize. Here you go. You got a promoter that the RNA polymerase might recognize as being the start of an operon. And then just downstream, and that's an interesting word, that's the right word, there's a flow to what's happening here. A polymerase would sit down here and then flow down in this direction, right? That's the natural thing. Downstream, and that's the word that molecular biologists will use, downstream of the promoter is another stretch of nucleotides called the operator. So the lactose operon has a promoter, an operator, or an operon, or an operator, and then the 3 structural genes. Okay? The operator is a stretch of nucleotides that a regulatory protein can bind to. The regulatory protein is called the lac repressor. And that's what allows E. coli to decide, should I be expressing these genes or not? That regulatory protein is encoded for by a gene called LAC-I, I for inhibitor, regulatory protein. It, too, is close by that set of 3 structural genes. All 4 of those genes are relatively close to each other on E. coli's chromosome. So here's a little bit of a different look at what's going on with the lac operon. Let me remind you, I had you where I wanted you about 5 minutes ago, and then I digressed into all the lay of the land sort of stuff that's going on with the nuts and bolts of an operon. Let me remind you, it's a real bad idea to express the genes of the lactose operon unless there's going to be a return on that investment. It's a real bad idea to express the genes of the lactose operon unless lactose is around. If lactose is around, you'll get a return on your investment. If it's not, don't bother. You're wasting your time and your energy. So how's this going to play out? You got your RNA polymerase here that recognizes a promoter, then wants to move downstream and start making an RNA copy of the coding sequences for those 3 structural genes. Beta-galactosidase, well, here they don't even have Z, Y, and A. Beta-galactosidase, the membrane permease, and then transacetylase. 3 genes that need to be expressed. And you got this operator. So look how this plays out in the real world. There's an important job that needs to be done. E. coli needs to be able to respond to a stimulus in its environment. What are we going to start with? Let's start with there's no lactose in the cell's environment. This is an important piece of information for E. coli. You know why, right? Because if there is lactose, it's ready to write out that check for 100,000 ATPs to make the proteins that'll let it cash in on the lactose. But if there is no lactose, it's not writing that check, right? So look what happens when there is no lactose. When there is no lactose, the lac repressor protein it's a protein that can seek out and find a specific string of nucleotides, about 20 nucleotides, that occur only once on the entire 4 million nucleotide long E. coli chromosome. The nucleotides of the lactose operon's operator. When no lactose is around, the lac repressor seeks out those nucleotide sequences and it sits down on them. And it sits down on them hard. Okay? The lac repressor protein, if there's no lactose around, sits on the operator, and the RNA polymerase, which was very interested in the promoter, can't get past it. The repressor sits there, it turns back to the RNA polymerase, and it says, none shall pass. You can't get by me. I'm not letting you transcribe anything else. Say no more, right? That turns out to be a darn effective strategy for shutting off the expression of all 3 of those genes. If that protein is hunkered down, if the lac repressor is hunkered down on the operator, you're not going to express the genes of the lactose operon. See where it's going, too, right? I'm sure. You must. It becomes obvious, I think, at this point. What else has to happen? It turns out the lac repressor is pretty well described as being I think, let's call it a 2-trick pony. It can do 2 tricks, but it can't do both tricks at the same time. One of its tricks is it can recognize and bind to the operator of the lactose operon. And when it does, it prevents RNA polymerase from transcribing the operon. What do you suppose its second trick is? It can also bind to the sugar lactose. It can't do both tricks at the same time. It either sticks to the operator or it sticks to lactose. If there's lactose in the cell, if there's lactose in the air, lactose tickles the lac repressor and causes it to come off of the operator. And what do you suppose happens then? RNA polymerase no longer has that roadblock there, and it's off to the races, making an RNA copy of the 3 structural genes that ultimately get translated into 3 proteins that ultimately allow E. coli to metabolize lactose. You must be in awe, right? It can't just be me that thinks this is incredibly cool. If you don't think this is incredibly cool, you've been dozing off, you've been texting, you've been distracted. Come back. This is simply awe-inspiring, amazing stuff. It's as simple as that, something that's so hugely important to E. coli. The decision as to whether or not it's going to blow 100,000 ATPs hangs entirely upon whether or not that repressor is bound to the operator or not. If lactose is around, it comes off. If there's no lactose around, it sits on the operator and it stops the expression from occurring. Logical, simple, elegant, totally effective. At a molecular level, you guys now see, if you haven't known this before, you guys now see how it is that E. coli does one of these fundamental things we consider for living things, being able to respond to their environment. It all depends on the function of this one protein, the lac repressor. And it gives rise to the perfect, the obvious, the necessary response to that stimulus from the environment. So when lactose is around, it tickles the repressor, changes its shape, allosterism, right? We've talked about that before. Did I mention to you guys that structure and function in biology are kind of interrelated? Oh yeah, it's come up. All right, so once again, you see an allosteric effect, how changing the structure of a protein can change its function. When you get lactose around, the structure of the lac repressor protein changes. It can't recognize the operator anymore. RNA polymerase zooms through and makes an RNA copy. Ribosomes are sniffing along at the butt of the RNA polymerase, and they're translating away, making those 3 proteins that will allow E. coli to metabolize lactose. This is one of those drop the mic, walk away kind of moments, because it's just not going to get better than that. But, you know, I got news for you. It does get a little bit better. That's not even the whole story yet, but this is an awfully cool story right here, right now. How about that? So that's the lac operon, or a big chunk of the lac operon. Okay? You know, in the student evaluations at the end of the semester, I read through those like it's Christmas Day, right? And I'm opening up my presents. I love getting the comments from you guys, the good and the bad. I learn a lot from looking at the student evaluations. Quite often I'll get a comment, or I'll get some comments that say, this Crane guy makes really simple stuff complicated. And I think they're talking about stuff like the lactose operon. Let me underscore to you guys, I agree. This is simple stuff. If you're thinking this is complicated or tricky, or it's just because I've done a really bad job of communicating it to you, right? When you come right down to it, this is the way to solve E. coli's problem. There is no good alternative. There is no logical alternative to this. If you were given, you know, the homework assignment, okay, you're all gods, right? And your homework assignment is to use your godly powers to design some form of life that does a better job at responding to the presence or absence of lactose. This is the only answer, right? Nobody else comes up with a better answer than this. It's simple, it's elegant, it's the way to do it. If you're not sure about how this is working, it's because you haven't just stopped and thought about it a little bit. There's no better answer. This is the logical, this is the best way you could respond to the presence or absence of lactose. So, you can test your understanding of what I've been talking with you about, right? All this is well and good, you know, cigar smoke and cognac sipping here. Oh, yeah, of course, that's how it works. You can put some rubber to the road for yourself and see if you know what's up with the lactose operon by asking yourself a question like this, right? It's part of the PowerPoint presentation. You can ponder it. with some cognac and a cigar later today, right? What do you got to do? The way that biologists often think about these sorts of things is they think about, what do you got to do to mess this up? How do you break this, right? And so, one way you might break it is like, what could you do that would mess up the system that I've just told you about in such a way that you'd make the cell unable to metabolize lactose, unable to get those ATPs out of lactose in its environment? What could you do? What do you think? A mutation to the LAC-Z gene, the beta-galactosidase gene? Mutation here means messing it up, right? How about a frame shift mutation? How about a premature stop mutation? Something that just really messes it up. That protein's not going to work. If you mess up the beta-galactosidase gene, guess what? You're not going to be able to metabolize lactose, right? Because I just told you messed up the protein that's the star of the show for metabolizing lactose. You could be clever here, and you could say, what if I messed up the permease gene, the LAC-Y gene? That would work too, because even though beta-galactosidase was around, if you can't get lactose from outside the cell to inside the cell, you're not going to be able to metabolize lactose. And if you can't keep the lactose inside the cell once it's inside the cell, if it goes out the same way that it came in other words, if you mess up the transacetylase gene, that also will do a number on your ability to metabolize lactose. So there's a couple of ways that you might mess this whole thing up. What about a mutation in the LAC repressor such that it can't recognize the operator? Right? So I've told you the LAC repressor is a two-trick pony. One of its tricks is it can bind the operator. The other, it can bind lactose. I'm telling you now, I've messed it up so it can't do its first trick. If it can't do its first trick, what's going to happen? You'll metabolize lactose all right. It's just you won't ever stop trying to metabolize lactose because the repressor can't sit on the operator and set up that roadblock if it can't bind to the operator. So whether lactose is present or not, you're going to be expressing those genes. So that doesn't destroy your ability to metabolize lactose, but it messes you up in terms of your energetic efficiency. Different kind of problem, but a problem nonetheless. What if you change the operator? Remember what the operator is? The operator is that string of nucleotide sequences that the repressor binds to, about 20 nucleotides. It only shows up once in the entire circle of E. coli's chromosome. If I change those 20 nucleotides so that the repressor can't bind, the repressor can't bind. If the repressor can't bind, I'm going to be expressing those genes whether lactose is present or not. The same problem as the second problem. It's an energetics problem, but I'm going to be metabolizing lactose. The question here is, can I metabolize or not? So it's a problem. The problem is not that you can't metabolize lactose. What if I mess up the LAC repressor's second trick, such that now the repressor can do its first trick, bind to the operator, but is now insensitive to the presence of lactose? What's going to happen there? The repressor will sit down on the operator, set up its little roadblock. You can throw as much lactose at it as you like. It doesn't recognize lactose anymore. I mutated it, remember? The roadblock is there whether lactose is there or not. I'm not going to metabolize lactose. That satisfies the condition, I suppose, of this question. I can't metabolize lactose, not because of any problem with the genes, but rather a problem with the gene that regulates the metabolism genes. All right. We're in the home stretch. You're going to enjoy the last 6 minutes here today, guys. Just you're going to have fun. Brace yourself. It's good. So the LAC repressor is the first, and it's the classic example of negative regulation of gene expression. It's negative because when the protein is doing its thing, the LAC repressor, when it binds the operator, it stops the expression of genes, right? It's a downer. It reduces gene expression. It's a negative regulator. The LAC repressor protein is a negative regulator. It's the classic example of negative regulation of gene expression, okay? And you can see from the outline, and I think even without the outline, you would have guessed that if there's negative regulation, there's probably positive regulation. What do you suppose positive regulation is all about? I'll tell you. Positive regulation means that when the protein binds to DNA, it helps RNA polymerase make an RNA copy of a gene, right? Check it out. Get ready, right? The lac operon has two switches. One switch that's a negative regulator and one switch that's a positive regulator. I've spent the last 15, 20 minutes talking with you about the negative regulator. I'm going to spend 4 minutes now telling you about its positive regulator. Okay? Because here's the thing. It turns out that in the energetic, economic sort of consideration that's going on here, there's two questions that E. coli needs to answer before it expresses the genes of the lactose operon. The first and the most obvious, I suppose, is there lactose around or not? If there's no lactose, it's just stop. I don't need to ask anything else. I'm done, right? But there's a second question that you guys may not have occurred to you yet. And the second question is, as far as sugars go, lactose is fine, but it's not E. coli's favorite sugar. Okay? You know what E. coli's favorite sugar is. It may not have occurred to you to have put it in those terms before, but you know what it is. It's glucose. If there's glucose around, E. coli really doesn't care if lactose is around or not. It's going to go through all the glucose first. It's the bacterial equivalent to this problem. Let's say that I have a big bowl here with me today of day-old cold broccoli. Maybe even make it a couple days old, like Thanksgiving old cold broccoli, right? Left over from dinner, and some nice, fresh, hot, Krispy Kreme donuts, right? Now, there's some good nutritional value. There's lots of things to be said about the broccoli, but I'm pretty confident, aside from those of you who really want to be contrarians and make a point, if I have two bowls, one full of fresh, warm donuts and one full of cold broccoli, nobody's going to eat the broccoli until the donuts are long gone, right? And you'll probably wait. We'll get locked up in here for a day or two before somebody says, okay, I'm getting hungry. I'll go for the cold broccoli. The cold broccoli is lactose. The donuts are glucose. If E. coli is in a world where it's awash with both donuts and broccoli, it's going to go for the donuts every time. If there's glucose around, it's not going to express the genes of the lactose operon. And you know how it pulls that off? It pulls that off with a positive regulator. The positive regulator is a protein called cyclic AMP receptor protein, CRP, cyclic AMP receptor protein. You know what AMP is? We've even talked a little bit in this class about how cyclic AMP can be a messenger. AMP is adenine monophosphate, so we're scraping the bottom of the barrel when you're down to a lot of AMP. It means the ATP has been changed to ADP, the ADP has been changed to AMP. Energy levels in the cell are getting pretty low if you've got a lot of AMP and cyclic AMP inside the cell. When energy levels get that low, cyclic AMP tickles a protein called cyclic AMP receptor protein. The cyclic AMP receptor protein changes its shape, allosterism, structure, function, once again. And when cyclic AMP is bound to the cyclic AMP receptor protein, it binds to some nucleotides that are found as part of the promoter of the lactose operon. It only binds then. And it's only when the cyclic AMP receptor protein has bound that RNA polymerase sits on the promoter of the lactose operon. And it's because, and I told you there are 13 nucleotides that RNA polymerase is looking for. The promoter of the lactose operon has only 10 of those 13. As far as promoters go, it's a crappy promoter. RNA polymerases will give some partial credit, but when you're off by 3 out of the 13 that it's looking for, it's not that excited. It'll sit down there, but it doesn't really want to do anything with it. Cyclic AMP receptor protein. It's the equivalent of having like Heidi Klum at my Christmas party. Suddenly, everybody wants to come. All the guys, at least, want to come to my Christmas party. Brad Pitt, whoever, for the women. You know, cyclic AMP receptor protein makes this a place RNA polymerase wants to hang out. When glucose levels are low, cyclic AMP receptor protein settles down at the promoter of the lactose operon. It recruits in RNA polymerases, and transcription can occur if there's no roadblock there. When glucose levels are high, cyclic AMP receptor protein doesn't sit there, and RNA polymerases aren't ever even going to have a chance to bump into that roadblock because they don't really think that's a very cool place to hang out. So the lactose operon is responsive to two things, and only two things, and it's in the environment of an E. coli cell: lactose levels and glucose levels. Lactose levels have to be high, glucose levels have to be low. Under those circumstances, you express those three genes. You write the check for the 100,000 ATPs that's going to let you get a return on that investment. If both of those conditions are not met, don't bother. Pretty cool, huh? Right? You're not going to have any more interesting lectures at all today, right? Just hang it up, go home. That's as good as it's going to get. But on Thursday, I'm going to tell you a little bit about another operon called the tryptophan operon, and then I'm going to talk with you about how eukaryotes play our trick of regulating gene expression. See you guys then.